Welcome to our podcast entitled Regulatory Perspectives in Precision Oncology Challenges and Opportunities for Track Inhibitors in Non-Small Cell Lung Cancer. My name is Dr. Justin Gainor. I'm the Division Chief for Solid Tumor Oncology at the Massachusetts General Hospital and Mass General Brigham Cancer Institute and an Associate Professor of Medicine at Harvard Medical School. And it's my distinct pleasure to introduce my colleague, Dr. Sanjay Popat, and let him introduce himself to the audience as well.
Hi, colleagues, and hi, Justin. My name is Sanjay Popat. I'm a medical oncologist at the lung unit at the Royal Marsden Hospital in London, England, and I'm also Professor of Thoracic Oncology at the Institute of Cancer Research, again, in London, England.
Great. And thanks so much for joining me. Obviously an exciting topic. And, I figure we'll kick things off just with an introduction for our audience on NTRK fusions. And I can kick things off. And, obviously the field of lung cancer has changed dramatically over the last, 15 years with the recognition that lung cancer isn't just one disease. It's really a distribution of various oncogenic drivers. And we now have regulatory approvals of so many different targeted therapies. And really this has transformed management of disease for many of our patients. NTRK fusions tend to be amongst the rarest. We sometimes say in our clinic, maybe you find one or two in a career, but these patients are out there and they really, really benefit from targeted therapies. In some of the retrospective series, it looks like 0.1, 0.2% prevalence. The nomenclature here can be a bit confusing. Right. So NTRKs are the names of the genes. So NTRK 1, 2, and 3 distinct genes. The corresponding proteins are TRK A, B, and C. And importantly, these drivers are mutually exclusive from others like EGFR ALK. And we also know that patients with NTRK fusions really have distinct clinical pathologic characteristics. We tend to see these in adenocarcinoma, never smokers, much like we do for many of our other targetable oncogenic drivers. Critically, they confer sensitivity to targeted therapies. And maybe, Sanjay, you can tell us a little bit about kind of what you expect to see with these agents in clinic.
Yeah, thanks, Justin. I think you're right. The field of oncogene-addicted disease has been transformed by the finding of oncogenic drivers. EGFR was the first one in 2004, and drugging a driver has been shown to cause rapid tumor shrinkage, which is durable, and that has really stood the test of time with all of the drivers that we've identified, particularly when it comes to NTRK fusions in NTRK 1, 2, and 3. And, as you said, I think understanding that these are three different genes with different fusions is important because people often think NTRK is one gene. It's not. And the reason it's important is initially we had two targeted agents, kinase inhibitors, directed against the NTRK fusion. Initially larotrectinib, and around the same time, actually entrectinib. And then more recently, we've had the approval for repotrectinib. And these are oral therapies, these are kinase inhibitors, these are daily tablets you take. And because of the rarity of the patients, the data that supports them have been pulled from phase 1 studies. And with larotrectinib, we've had the adult phase 1 trial, the pediatric SCOUT trial, the Navigate trial, and basket trial of patients with many diverse diseases, many different tumors, all of which have the common finding of oncogenic fusions in NTRK 1, 2, and 3. And what Dr. Dryland and colleagues initially reported was rapid and durable responses. So responses occurring in up to 80% of patients, lasting beyond 2.5 years on average. With entrectinib, we see very similar data. Again, this was pooled analysis from the ALCA-001 phase 1 trial, together with the Star Trek 1 and 2 phase 2 trials. Response rates, again, similar, maybe numerically slightly lower at around 60%. PFS, maybe numerically slightly lower, around a year or so, but still very effective therapies. We know, however, that we always get acquired resistance with kinase inhibitors, and this is a problem that we have. Alongside now comes the new player, the new kid on the block, repotrectinib, which is a next generation NTRK inhibitor designed to get into the binding pocket to overcome resistance. And what we see there is a high response rate, around 60%, very rapid responses. And in NTRK naive patients, this is very durable for over 2 years or so. But what's really important is if your patient has on-target resistance because of a gatekeeper solvent front mutation, in the solvent front mutation group, we again see resistance from this, with responses around 60%, duration of response 7 months. So very active drugs in this setting.
I think that's a fantastic overview of the clinical data, and I think it really highlights that the development of NTRK inhibitors, this wasn't done in isolation, right? It built off of the history and the biology that we've come to understand for oncogenic fusions. So, we had already known about ALK ROS1 fusions, right? And so we know that in the setting of these chromosomal rearrangements leads to constitutive kinase activation, as well as oncogenic transformation and this state of oncogene addiction. And that's really what's enabled us to develop targeted therapies and deploy them in clinic, even despite the rarity. I think the other thing that gets a lot of attention, and appropriately so, on NTRK is the implications it's had in terms of drug development, right? I think, obviously, as two thoracic oncologists, we tend to focus on lung cancer, but I think NTRK is also not unique, but one of the noteworthy aspects of this as a driver is it occurs not just in lung cancer, right? It occurs rarely in many common tumor types, and then it's more common in some super rare tumor types, right? And so, this has really spawned this idea of, when you're developing an agent in such a rare molecular alteration, and you've already alluded to this idea of kind of basket trials, right, where you take patients regardless of tumor type who have a given alteration, and they're eligible for a study. And so this idea of tumor agnostic clinical trials and also tumor agnostic regulatory approvals, which this was really one of the first examples of tumor agnostic approaches. And so I think one of the benefits of that approach is, one, speed, right? Speed and feasibility. I think if we just tried to do an NTRK specific trial in lung cancer, we'd still be waiting for the data, right? So I think this allows us to test the activity of these agents in a rapid way, in a very pragmatic way. And it can also give us biological insights into the driver itself, which is, if we're seeing high response rates across tumor types, we recognize this is a shared vulnerability, across tumor types. So I think that's another positive aspect of these basket studies. And then lastly, I think it also gives us an ability to look at safety in a larger patient pool and across disease types. So I think that that is another benefit of the basket approach. Um, Sanjay, do you think there are limitations of this approach, or kind of limitations of the data that we have thus far?
Yeah, one of the problems that we've got with this approach is really understanding the granularity of what drugging NTRK is bringing to the disease in the wider context, right? We know what it's doing to the individual, but it makes it very difficult to understand what's going on in in the overall group. So, for example, you know, if I've got a group of, nearly 30 different tumor types, ranging from adult tumors, mesenchymal tumors, epithelial tumors, lymphoblastic tumors, if I've got, for example, pediatric tumors, where do I bring this drug therapy into the patient's paradigm? Is this the first treatment that they ever have? Do I use this in a curative setting? Do I use this in a late metastatic last line of care, or somewhere in between? And I think it's that level of granularity which becomes a problem. So the heterogeneity of the study populations is particularly problematic, particularly when you've got pediatric patients on one side of the argument and you've got elderly patients with lots of comorbidities on the other side of the argument. The other issue is, so look, you get very good shrinkage and you get shrinkage for a reasonable period of time. And after that reasonable period of time, this data needs to be in the public domain, right? Because it's making a difference. But of course, we want to know what longer term impact it has on patients, both in terms of efficacy and safety. So, is it really making a difference on survival? Are people living longer as a consequence of this new therapy? You'd have thought they probably are, because it's a highly effective therapy and it's keeping the cancer under very good durable control for a pretty reasonable period of time. But actually, is it making that much difference? Because in some tumor types, they could be very, very sensitive to other um, types of treatment. Now, whether you'd rather have a track inhibitor or chemotherapy, I know what I'd rather have, right? But there is an argument to be made at the population level in terms of the payer's viewpoint in how that is perceived. And then we have to think, well, the data that we have are essentially surrogate endpoints, right? Because the gold standard is overall survival, and we're never going to be able to do randomized studies in this setting. So these are the issues we need to think about when we're interpreting these studies. But Justin, I guess they're going to cause issues down the regulatory pathway where the regulators start thinking about this.
Yeah, and that's a great segue, because obviously practicing in two different regions of the world, I think it does highlight now. I can maybe weigh in on the U.S. perspective, and I would love your thoughts on the U.K. perspective. I think that, as I already alluded to, you know, the the there were a couple features here. One is the unmet need, right? But before this, patients with NTRK fusions, particularly with lung cancer, we were just left with chemotherapy, checkpoint inhibitors, right? So clear unmet need. Um, and I think that the FDA took a very progressive view here of this idea of a tumor agnostic approval, basket studies. They were quite small, right? And really anchoring the approval on objective response and durability of response. To the point you were making earlier about different tumor types and some of the uncertainty, not like a time to event endpoint like PFS and survival, just given that heterogeneity. Um, and post the approvals, right, with some post marketing commitments, but notably no, you know, need for a randomized study, and which I think is appropriate. We'd still be waiting, but maybe you can give us some of the UK perspectives.
Yeah, so in the UK, we and in Europe in general, right, we have a regulatory process and then we have a reimbursement process. And I think that's slightly different to the US, right? Because the sort of regulatory process is aligned to the reimbursement process in the US. So from a UK specific process, the UK is now outside the European Union from a regulatory perspective. So Before Brexit, we were reliant on EMA as we were in the European Union. And so the companies would then submit their dossier to EMA, that would then review the dossier in terms of the risks and the benefits. And if the drug was granted an approval, it would be granted from EMA, and that would flow through to the UK. Post Brexit, the UK has its own competent authority, the MHRA, who is now able to issue a marketing authorization for a drug targeting NTRK. Now, the key issue is how do you then get that in the setting of a rare disease? There are several ways that might happen. If you've got a drug which is extremely exciting, something that is looking very effective, as these drugs have been, there is the opportunity now post Brexit to align with the FDA, because the FDA has a program called Project Orbis, which allows the regulatory FDA submission to then be submitted to other partner regulatory authorities in different countries participating in Project Orbis, and the UK is one of several of these, such as, for example, Swissmedic. And that allows a very rapid approval in the UK setting. Now, the manufacturer may decide that for whatever reasons that that's not the route that they want to go down. There is what we call a direct mechanism of submission, so the manufacturer can submit through a number of different routes into the MHRA directly. But there's also something called the EMA reliance route. So often what might happen is the manufacturer will say, well, look, actually, we're not going to go directly to the MHRA. We're going to submit our dossier to the European regulator, the European Medicines Agency, EMA, who would then issue their approval, and the UK approval is then reliant on the EMA approval. This is historically what used to happen. And in this setting of when we have small data sets, usually there is a specific post-authorization obligation. And we've seen this in many single-arm studies. The data set would be submitted perhaps on 60, 70 patients worth of data. The regulator would grant a conditional approval and then say, well, that's fine, that's fair enough. You either now need to run a randomized study, which we know in NTRK cases is going to be impossible, or you need to have a larger cohort to continue to accrue this data with longer follow-up so we can be more sure of where that response lies, what the duration of response lies, what the adverse event profile is like, and then the relationship between the two. And after that, after the label, all the countries in Europe have a reimbursement step. And in the UK, and specifically England, this is a body called NICE, which determines the cost effectiveness using something called incremental cost effective ratio, an ISA, which looks at the quality of adjusted life years gained for implementing the new therapy. And each country within the European area has its own health technology assessment, HTA, mechanism for assessing the cost effectiveness of bringing that drug in. I think that's slightly different to the US system, isn't it, Justin?
It is, and I think you draw an important distinction there. As part of the regulatory approval, it's not linked to cost effectiveness. So those are disentangled but between the two. So it's really, is the drug safe and effective? But you're right. I think it is a distinction.
And in the UK, there are a number of different technical issues to this. So for example, when these drugs are being evaluated by NICE, there are a lot of uncertainty that's driven by the longitudinal outcomes, and the longitudinal outcomes drive the uncertainty in the cost effectiveness. And that creates a problem, because when we've got more uncertainty into what the longer term outcomes are, there's more uncertainty as to what is cost effective. And this is particularly problematic for rare indications. So, for example, we're in the UK at the moment, entrectinib was originally approved in 2020 under a conditioning funding agreement, and in 2025 it came up for a re-review of funding, and this year the manufacturer did not provide an evidence base for that, and so entrectinib funding for NTRK positive tumors was withdrawn by NICE. And when we look at the regulatory documents, in that 6-year period, only 12 cases were registered in the national database for using entrectinib for an NTRK positive cancer, highlighting the rarity of this. In larotrectinib, the renegotiations after the 5-year period are still ongoing. So larotrectinib was originally reimbursed in 2020, and in the public domain documents that we see, only 60 cases have, for 60 cases have as larotrectinib been used in England for NTRK positive cases, highlighting some of the challenges that we have with health economics. And the other thing we need to think about as physicians, the local health authority will also put additional restrictions on this. So, for example, NICE will say, okay, that's approved and funded for ECOG performance status 1, but then NHS England will say, oh, well, it's only ECOG performance status 0, 1. You can only use it last line of therapy, and you have to have patients with these additional restrictions, such as liver or blood function requirements, no active brain metastases, etc. And these, often cause problems with the inequity of access.
I think that's a great point, and actually a nice segue. I would like to pivot a little bit to disparities in access to treatment, and I think you just highlighted several of them, and I think this also starts with access to testing, right? It's not just access to the drug is the second part, right? Access to testing is certainly a major barrier. We know for certainly lung cancer, but many other tumor types, you know, um, to find NTRK, there are a couple of different molecular tests, but as you start screening for, you can't just do single assays for each gene, right? Especially with the number of tests that we have. So next generation sequencing tends to be, preferred modality and testing for all drivers simultaneously. Sometimes fusions can be tricky to identify with some platforms. I think for NTRK in particular, it does suffer a bit from the rarity, and you highlighted the number of cases, identified, since 2020, and that certainly is a concern. But I think partnering it with, hey, we have to be testing for all these other alterations, this really underscores, this is easy to just do the broad testing. Um, and so I think that is really important, and yet we have people still not getting tested. I recognize it's multifactorial, right? Sometimes the tissue is not adequate. Sometimes we have to think about a repeat biopsy, but I also think that blood-based testing has made this much more straightforward, knowing that if there's not enough tissue, that if we find something in the plasma, we can certainly pursue treatment. Other things that you think I'm missing in terms of barriers to testing?
Well, I think you hit the main ones, and the rarity is a particular problem, right? Because folk, forget that it's relevant. I mean, that's one of the key issues. The plasma testing is a really important one. In England, we're lucky enough to have a plasma first program. So when the patient hits the respiratory clinic, pulmonology clinic with suspicious symptoms or a suspicious CT scan, they're actually funded to have a ctDNA test at presentation, which is very forward thinking. But Justin, as you know, picking up NTRK fusions on ctDNA is challenging, right? Because the technical aspects and chemistry of ctDNA. And actually, as we know, RNA sequencing is problematic as well, right? Because you have to have a certain amounts of tumor. Sometimes the RNA fails because of processing issues. So there are multiple, reasons why the test can be funded, but actually the test is not really done or done to the adequate quality. And these things take time as well, right? NGS takes time, and so getting these results is problematic. So there are many different problems with this. We're trying to get reflex pathways to, overcome these limitations. Luckily, I think we've got adequate resources, but some of the coordination between the path lab and the molecular lab, I think, could be a lot better. And we just need to also ensure we have adequate genomic literacy. I do think one of the big issues is that folk may not know that they've got a NTRK fusion in that report. So adoption of RNA-based testing is really critical. Having clear reports is really critical. Adequate education for our oncologists and the wider clinical group is really fundamental to ensure that we're getting rid of all of these different bottlenecks. What do you think the future's looking like, Justin, for this and other rare biomarkers in lung cancer and other diseases as well?
Yeah, I would think in two aspects. I think on the scientific aspect, right, and then on the regulatory side, right. Biologically, I think as sequencing technologies evolve, now this long read sequencing and other things, I think we will continue to make novel insights into new therapeutic vulnerabilities. As we make these insights, though, these are likely to find increasingly rare populations. And so I think we need to think more dynamically from a regulatory perspective because we can't do big randomized phase 3 studies for all of these. And then we have to start thinking about, even, is it possible to do single arm studies, or can we augment that in some way? That is, can we create synthetic control arms, digital twins? Can we leverage real world data? I think all of these things are on the table, especially as our health systems, particularly in the US, are increasingly integrated with our EMRs. And I would say as the testing companies start collecting more outcomes data, it really does help facilitate some of this real world evidence generation. And I think that's particularly important for alterations like NTRK, where a single institution may only have a few patients, but then you look at a system-wide level, at a country-wide level, at a global level. I think that can really help with the evidence generation. How does our regulatory structure evolve also as the biology evolves? And so, like, how do you think about that? Now we have, you mentioned 3 track inhibitors, right? How do you think, is there a role for newer track inhibitors, and how do we evaluate them within a regulatory context? And now that one could point to say, hey, there's 3 drugs, is there still an unmet need?
Yeah, great questions. I mean, yeah, definitely, we've got an unmet need because patients are still dying of NTRK positive lung cancer. Right? I mean, that's the bottom line. Our drugs are good, but they're not that good, right? We need better NTRK inhibitors. We need NTRK inhibitors with a better range of side effects, make them tolerable. They're really problematic, NTRK inhibitors. And we need NTRK inhibitors that can overcome diverse mechanisms of resistance, both on target and think about how we combine them with other drugs as well. But more than that, right? We need to think really about rare biomarker-driven subsets in cancer, right? How do we achieve regulatory reform? Because the regulatory system fundamentally, maybe less so in the US, but fundamentally in Europe, is still very much based on a randomized phase 3 trial, right? That's really what the regulators are looking at. That's just not going to be possible, and if I have one rare biomarker which is doing wonderful things to a super rare population, it still creates big issues with reimbursement. So our reimbursement system is also in a very old-fashioned setting aligned to having something comparable that we know what the cost is over a longer period of time. So we need to think a bit more clever. We need to think about accelerated approvals. We need to be comfortable about single arm studies to get the approvals in to then generate wider data, as you have done with the FDA system. I think that's really important in Europe to then allow a greater amount of flexibility when it comes to reimbursement. So some things are happening, with the wider political agenda that we've seen globally. We've seen some lifting of the thresholds with NICE, driven by some of the economic changes we've seen in the US recently. And we've seen some difficulties with NICE and the UK environment. We used to have something called the end of life criteria, which lifted the quality threshold from 30K to 50K, allowing greater spend for some, cancers. But that's been eradicated, and now we have something called severity modifiers, which essentially modify the funding index from 1.2 to 1.7, a weight that they give to the quality. And this is causing some problems because there isn't debate as to which level of severity modifiers your particular disease is suitable for. So I do think there's still a lot more work to be done, and hopefully the HDA agencies and the regulatory agencies, alongside with clinicians, can coordinate a bit better. But I'd be interested in your thoughts on that.
Yeah, I completely agree. I think this has been a great discussion. I concur that we need greater alignment, right, between regulators, sponsors, clinicians, testing companies. That this conversation has really highlighted that, and I think that especially as we think about expanding targeted therapies to other oncogenic subtypes, not just within lung cancer, but more broadly across oncology. So this has been a phenomenal discussion. I really want to thank our audience. I really want to thank Sanjay for joining and for his expertise. And just one final reminder to our audience to complete the feedback form would be really helpful for us moving forward. And so with that, I'd like to thank you again.